everyone. My name is George Huster. I'm a third-year PhD student here in Newcastle, and I'm here to talk about some of my research on coral proteomics. Now, that probably doesn't mean a whole lot to most people here, but hopefully by the end it will mean something. So, let's start from the beginning. What is coral? Well, coral is alive, and it is composed of two organisms living in symbiosis. You have the coral itself, which is an animal, and inside the coral cells live an algae. And this allows the coral to both perform photosynthesis, like a plant, but also filter feed off of debris in the water. And they are incredibly important ecosystems. They are the foundation of coral reefs and which are home to 25% of all marine life, despite being less than 1% of the ocean floor. However, these lovely ecosystems are very much under threat. As ocean temperatures rise, the symbiosis relationship between the coral and the algae break down, and this causes the coral to lose its pigmentation, a process called bleaching. And if corals stay bleached for too long, this can lead to them dying. However, as you can see in this picture here, there is some natural level of variation between how tolerant corals are to heat stress. You can see even within the same species, there is these corals right next to each other exposed to fairly similar conditions. Some of them have bleached completely, some of them are doing completely fine. Which means there is hope for selective breeding projects to further enhance what is allowing these corals to survive. So that brings me to what do I do? And so I researched how corals produce different proteins in response to heat stress, a process called proteomics, so the study of proteins. And proteins are very, very important because they govern a lot of biological functions. They can help hold the cell together and give a cell shape. They can catalyze biochemical reactions. They can act as part of signaling networks. So research into how the abundance of proteins change is very, very enlightening. And I specifically look at how our line of selectively bred corals responds differently to heat stress. So for some background, a lot of our research is based out of Palau, and we sampled some coral off of the reef there, and we sorted them by how heat tolerant they were through a heat stress experiment. We sorted them into two piles, the survivors and the not-so-survivors, termed the HF0 and the LF0. We then selectively bred these two groups within each other to produce the next generation, the HF1s and the LF1s. So all you have to remember is the H stands for high tolerance and the L stands for low tolerance. In total we have 19 LF1 individuals and 34 HF1 individuals, and these right here are all of the offspring are all of the experiments I'll be subject of the experiments I'll be talking about today. So, as these corals were growing up in what we call the nursery, a big steel table-looking thing sunk to the bottom of a bay, they experienced a relatively mild but prolonged heat stress event. So this resulted in none of the corals dying, but some of them bleached, about half of them bleached. And so we collected tissue samples from all of these corals, and we extracted all the proteins, quantified them, and we compared which proteins were differently expressed between the two groups. And the first thing we wanted to look at was heat shock proteins. These are very, very highly conserved proteins. Everything alive, basically, ranging from bacteria to us, all have these heat shock proteins. They are responsible for many, many functions, chief of which is to make sure that during heat stress, they make sure all the other proteins are repaired and doing fine. And all of the literature thus far has shown that the more heat shock proteins you have, the more heat tolerant you are. And we saw that the heat shock proteins were significantly more abundant in the LF1 population. Wait a minute, the LF1 population? That's clearly something's gone wrong. So our initial hypothesis, obviously, was more heat shock proteins, more heat tolerance. And we would expect the exact opposite result. But that is clearly not the case. After triple checking I didn't miss a minus sign in my coding, we came up with a new hypothesis. Remember when I said earlier that the heat stress event was relatively mild and prolonged instead of what the parents were bred for, which is very intense, severe, nearly fatal heat stress events. So our next hypothesis was that the mild heat stress was causing the low heat tolerance individuals to get stressed out, but not the high tolerance individuals. And this was likely caused by a buildup of reactive oxygen species. And reactive oxygen species, for short, are these damaging molecules that are produced by a variety of cellular processes, and they can they go on to cause non-specific damage to all parts of the cell. And indeed, we see all of the repair mechanisms associated with reactive oxygen species damage is elevated in the low heat tolerance individuals as well. So this then allowed us to produce this sort of model where under the mild heat stress, we get a buildup of reactive oxygen species which then can then go on to cause all of this non-specific damage to lipids, proteins, DNA, and the high tolerance individuals are more tolerant by virtue of preventing the damage before it has happened, whereas the low tolerance individuals are busy firefighting the damage that has already happened. They're trying to contain the damage. And to confirm this, we then looked at a follow-up experiment on the same individuals, and instead of the mild heat stress, we put them through full-blown heat stress, and we saw the results reverse as we were expecting. So the previous differences were basically disappeared as the heat stress went from mild to severe. So the take-home message of the day, I guess, is so under mild heat stress, the low heat tolerance offspring behave as if they are under full heat stress, and the high tolerance individuals are more tolerant by virtue of preventing damage before it has happened. And therefore, this suggests that heat tolerance, quote unquote, which is what we're breeding for, is two-tiered with you need good heat damage prevention, but also damage control after the damage has already occurred. Thank you very much. Any questions?